Welcome back. This is part 2 of chapter 12, video analysis. Let's share the screen and begin where we left off in the previous video. We were looking there at microsatellite instability resulting in expansion or contraction of regions of DNA. So microsatellites include satellite DNA. Let's move on to continue the same process of replication errors, in that during DNA replication, that is a very vulnerable time for the DNA. As you can see in the figure below, 12.8, if the DNA replication process stalls during DNA replication because of some artifact that it cannot skip over in the template strand, the remaining template strand is still held single-stranded. And any kind of break across there will separate this fragment of the chromatin from this fragment. And suppose the centromere is over here, then you will get this fragment of DNA floating away. And that will be equivalent to a double-strand DNA break. So think about that. That's very important. Figure 12.9 goes on to show us two alternative mechanisms that can also result in DNA damage. The first one, the panel A, shows us what happens when the glycosidic bond holding the base to the sugar is broken. We get a AP site where there is no base, and that can cause issues with DNA replication and also in transcription. The alternative mechanism is where you get deamination, where the amino group is somehow lost itself, and that can result in the original base changing identity. The most serious of these are the top two. So here we have cytosine, and when it loses its amino group, it becomes uracil. Now, uracil is easy to identify because it doesn't exist inside DNA, but uracil does exist in RNA. So that should be pretty easy to identify. Adenines, they can also become deaminated, and they can form hydroxanthine, which is also easy to identify by particular enzymes and repair mechanisms. Likewise, guanine, when it gets deaminated, it forms xanthine. So these three are easy to identify when present along the DNA, as we'll see later. Sometimes cytosines become methylated as part of a normal process of epigenetic control. So 5-methylcytosine is a normal constituent of DNA at many locations, but when it becomes deaminated, it resembles thymine. And thymine, as you know, is a normal constituent of DNA, and that can cause huge problems in repairing that kind of base alteration. And we'll come across mechanisms that try to minimize that later on. So moving on to the next page, it's important to realize that one of the most dangerous things inside the nucleus of a mammalian cell is the free radicals produced by the systematic breakdown of molecular oxygen. So doing oxidative phosphorylation, I nearly forgot the word there, doing oxidative phosphorylation, various species of free radicals are produced and other reactive chemistries that have to be regulated and controlled. But they sometimes escape the control mechanisms or the compartments in which they are generated and interfere with other compartments. And that can cause chemical alteration of DNA bases. Moving on to look at alternative mechanisms that are the cause by this type of misbehaving oxygen-free radicals. We have oxidation that can result in the formation of weird bases. So in this case, we have deoxyguanosine, so this is G, and the G then becomes oxidized, unfortunately, because of these free oxygen radicals. You can see there's a shift in the chemistry in the structure of the guanosine. And that can cause it to mispair with its partner strand, as indicated here. So normally we have A with T and G with C. In this case, we have A base pairing with G because of the shift in the capacity of forming hydrogen bonds across the complementary base pairing. So we have 2 hydrogen bonds between an A and a G instead of the regular 3. So this 8-oxo-deoxyguanosine, keep an eye on that, please. You're going to see that again and again in future discussions. That's one of the most dangerous altered chemistries that exists inside our DNA. In the bottom panel here, we can see that we have the same methyl cytosine that we looked at in the previous figure, and this is just a sequence of steps that leads to the formation of thymidine, right, or thymine. Okay, so this is just an intermediary, and of course, this is caused by, in many cases, these reactive oxygen species, or ROSs. The next figure, figure 11, simply shows us the relationship between the size of a mammal, its rate of respiration, and the amount of oxygen consumed, and therefore the amount of reactive oxygen species that could be produced. So if you look at small mammals, have high oxygen consumption, therefore maybe they produce more free radicals. That may limit their life expectancy. Whereas larger mammals, they have slower metabolisms, and therefore they produce more controllable levels of ROSs, and maybe that increases their life expectancy. So there could be a correlation right there, depending on the rate at which oxygen is assimilated into the body. We next go on to look at how genomes are under occasional attack from outside mutagens and their metabolic products, right? So in this particular case, we're looking at physical radiation coming from the outside, such as ultraviolet radiation. So the most common artifact produced by UV light is the formation of pyrimidine dimers, right? So here we have 2 bases on the same strand of DNA. They get covalently interlinked, and that can cause the replication machine to stall, as well as repair systems. And that can then lead to artifacts. Alternative chemistries can also exist due to the same type of radiation. And in each case, we can see that there must be mechanisms responsible for correcting these type of mistakes. In higher mammals, such as ourselves, the mechanisms seem to differ from those found in plants and lower eukaryotes, and indeed bacteria. And we'll discuss them at length as we get reading into this section. Alternatives from regular oxidative phosphorylation involve enzymes that are produced within the mitochondria in most cases, and they can also result in the aberrant formation of alternative chemistries. So in this case, we have cytochromes, and these cytochromes can act on normal chemistry that's consumed in the diet, and sometimes change that chemistry into a form not present within the body. So here we have a procarcinogen, a substance that is not known to cause cancer, but can be converted accidentally by the enzymes within the liver to forms that are very, very carcinogenic. So if you eat a lot of burnt food, you're going to be ingesting a lot of these types of chemistries, which in themselves are not dangerous, but because of the presence of enzymes inside your body, can be converted to very harmful substances. Cigarette smoke also introduces large quantities of these types of very poisonous materials. Okay. The next highlighted word here is DNA adduct. That simply means chemistry that binds to the surface of the DNA. In most cases, the bases. So we move on to see that. So here we have a BPDE, a very carcinogenic molecule, again produced in large quantities by cigarette smoke. And when it finds its way into the nucleus, it has a liking, I should say, for binding to guanine, and then it becomes a DNA adduct. And what it does is it prevents the DNA repair and replication machines from doing their regular jobs. Okay, believe it or not, alcohol also has an intermediary that can attack bases in DNA by binding to them and causing minor alterations that can then lead to more dangerous outcomes. So you can see here a normal G base in the DNA gets attacked by this molecule here. You can see it right there, added to the end. And then that can then react with amino acids that are present in the vicinity of the DNA, causing these DNA adducts. Okay, so drinking alcohol does increase the likelihood of getting cancer in those regions where the alcohol is present in high concentration. That'll be your esophagus and in your stomach, and to a smaller degree within your small intestine. This figure by itself seems to be complex, but it's really not. So let's systematically dissect it. So on the y-axis, we have a protein. P normally stands for protein. P53, which is the 53rd protein identified by this cancer lab. P53, as you'll see, will be one of the most common proteins we talk about throughout this course. So P53 mutational frequency. So this is a percentage, so 7% at the top, 6% for this graph, 16% for that graph. But the axis is exactly the same, right? So this is the percentage of cancers that had the P53 protein mutated in some fashion, okay? So we can see at the bottom, the codons, the use of different types of codons that are present. And the color coding here in the pie chart will help us understand that. So for the P53 gene, if this is the beginning of the actual coding sequence, and this is the end, you can see there's a couple of hotspots near the middle of the gene that are very, very susceptible to becoming mutated. So that's what this is saying. And now we can use the pie chart to quantify the type of mutations that are most common. And the key is given down here, right? So here we have a GC base pair being converted to a TA base pair, right? So the G is actually converted to a T somehow. Okay, so a G is a purine and a T is a pyrimidine. So is that a transversion or a transition? So that's what you need to find out by reading the text or going back to your Biology 2 textbook. And you can see here, we have also deletions and insertions. Other means other minor types of alteration in the DNA. But the color coding continues to look at various types of changes that can take place. So going back to all cancers, of which 15,000 were measured by this study, the most common type appears to be the yellow box, right? And that corresponds to a GC base pair becoming an AT base pair at these methylated CpG islands, right? These methylated islands. So that's the most common type of mutation found in all cancers. But we now do a subsection and just look at lung cancer in various alternative environments. So these are non-smokers. This is lung cancer in smokers, and this is lung cancers in people that live in coal tar country, right? Or they smoke coal for fuel. And you can see the numbers are quite small here, but these are reasonable. And you can see there's good correlation between this and this, and between this and this. However, here you can see many of the mutations are restricted to just a few sites along the gene for protein P53. And also you can see that the nature of the bar chart is beginning to change. So here, 3/4 of the changes caused by smoky tar are of the generalized G to A changes. So this is important. Why is it important? Because it gives us an idea of how that environmental agent interacts with our DNA. So this figure may look hard from the outside when you first come across it, but guess what? It carries a wealth of information that's very important in forming a true perspective on cancer. So please don't ignore that. Moving on rapidly, this figure again may look daunting at first light, but guess what? You can do it. You can conquer this. So look at the axis first. So here we have a scale. So 0.1 per megabasis 1 per megabase. So this is a logarithmic scale. That means it goes up in a non-linear fashion. All right. And what does this mean? Maybe these are the number of changes per every million base pairs, right? And down here we have buckets. So within these buckets, we have different types of cancers. We have pediatric cancers at the beginning, childhood cancers, and then at the end, we have more adult cancers. And also, as you live longer, you get exposed to carcinogens at a greater and greater rate the longer you live. And then we have another axis on top. And this one simply says the number of dots, i.e. the number of cancers that were examined to generate this particular graph, right? And then we have a key over here, which again talks about the conversions of a base to a different base. And then the actual bars are represented within these cancers. So let's start at the beginning very quickly. So there should be 109 dots here. You can't see them all, but they are 109 dots corresponding to the number of subjects that they examined in this pediatric blood cancer, right? AML. So it tells you all the abbreviations down here for the different types of cancers. Some of them are very, very common throughout this book. Others are only mentioned in specialized conditions like this. Regardless, it doesn't matter. We have the general trend, right? The longer you live, the more you get exposed to the environment. So here we can see that there are very few changes in the DNA because the individual is young and there's not enough time that passes. So these cancers are more prominent using small changes in the DNA, whereas these cancers appear to be correlated with large changes in the structure of the DNA. The only thing to note are the colors here are changing to more and more yellow as you approach this side. So this is melanoma, and that's caused by sunlight. You can see this is caused by diet, and these three are going to be the result of smoking. So the longer you live, of course, the more sunlight damaging effects take place. So here we have C to T mutations, very common, right, in melanomas. Whereas on this side of the scale, AML, in AML, we have a large portion of T's going to G's. So this is just telling you again that the biology of the individual, a developing child, versus a long-lived adult are going to impact the types of damage that the DNA sustains over time. That's all this graph is telling you, okay? Just to take that one step further, some scientists have correlated different types of environmental agents with cancer. So in this case, we have a correlation with aflatoxin. Aflatoxin is a very nasty chemical produced by fungi to protect themselves from other pathogens, right, themselves. So this is part of China, a province in China. And we can see right here near Shanghai, along the coast, there's a very high level of this type of cancer. So how does aflatoxin B1 in particular work? Well, again, once it enters the liver, the cytochrome enzyme, the P450, sorry, I was going to say P45, but that's something else. The cytochrome enzymes then don't know what to do with it, and they convert it to an intermediary. Now, the next step depends on whether this substance becomes carcinogenic or not. If it goes down this branch, then it becomes detoxified, and it has no carcinogenic or tumorigenic properties. So you're safe. If it goes down this pathway, then it's going to become a DNA adduct. It's going to bind to DNA and interfere with replication and repair. And if it goes down this pathway, then it's going to be detoxified into something that's safe. So the two brown molecules are great, but the red molecule, that's going to cause issues. So what happens? Well, it depends on the level of this. And it goes down this pathway. If there's high levels, then these systems simply get saturated, right? So depending on the load of aflatoxin in that environment, you can cope with small, tiny amounts because they get detoxified by these enzymes, but large amounts, they become DNA adducts. It's interesting when you read this that there are also there's also a correlation with the presence of a virus that is present in this part of the world. And that virus also causes, it's called hepatitis B. It also can have detrimental effects in terms of cancer. But the virus is present at equal quantities within that population, within that province. And the aflatoxin is present at a higher rate near the coast because of humidity. So you can see we can take away the impact of the hepatitis B from the population's baseline and come up with the remainder, which is due just to the aflatoxin. So eating contaminated or weakly stored grains is the most common cause of aflatoxin poisoning. Figure 18 simply gives us a listing of some of the most common exogenous mutagens that are present in food, particularly red meat, as it says. And one of the most common that enters our bodies, if you have a diet that's rich in red meat, is PHLP. And now this particular substance can be converted by enzymes in the liver to something a bit more dangerous, which is a DNA adduct. And then this goes and binds to the G bases on the DNA, and that can then stall replication and repair machinery. So minimizing your consumption of certain types of food will help mitigate some of the effects when it comes to cancer. So since we're talking about the mechanisms that the body uses to prevent itself from developing tumors, are there any specialized mechanisms? One of the most beautiful is indicated in this figure, figure 19. So here we have a patch of skin on the external surface, and the sun would then be beaming down from above. And here we have cells that are deposited in the living part of the skin, and they protect their DNA by forming these umbrella-like structures, which are known as a supranuclear caps. So there's a high concentration of melanin that's deposited at the north side of the cell, i.e. the side from which radiation is coming from the sun, and that acts as a barrier to harmful radiation hitting the nucleus, which is marvelous, right? So we find this. And of course, the greater the strength of the sun, the more of these caps are generated, therefore the darker the skin of the individual. Figure 20 gives us another example of protective agents throughout the body. So in this particular case, we have two entities. We have a substance called glutathione, and glutathione is GSH. It's a 3 amino acid substance, a peptide, and the middle amino acid, cysteine, carries a sulfur, and that sulfur is going to be very important. Then, in order to make use of this product, we need an enzyme, GST. What is GST? Is glutathione 5 transferase. And what it does, it performs a sequence of chemical manipulations which are designed to detoxify exogenous chemistry that enters the cells. Okay, so we can see in panel C that we can detect the presence of the enzyme in cells, but in cancerous cells, in this case the prostate, they have lost the ability to manufacture this enzyme. Therefore, they are more susceptible to exogenous chemistry. So it can be as simple as an enzyme missing that can lead towards tumorigenesis. So moving on to other definitions, they have another panel here which talks about individual differences between cancers and the heightened risk that we have, because we all have slightly different forms of genes, alleles, and they can empower us with different capacities. So that's going to be important for this too, right? Then we look at now the repair mechanisms that do exist. So most of these you should be familiar with from Bio 2. So we'll go through this relatively quickly, and you should have covered these in Bio 140. So as we continue to ask you to remember more and more examples, here are two more. So in this case, we have a G base in the DNA. It gets attacked by a alkylating agent. Alkylating simply means it adds an alkyl group of some type, right? So we here have an ethyl group. So this will then cause the base to misbehave when it comes to pairing with other structures in its base pairing complement. So that has to be fixed. So one way we learned in a previous example was the removal of the base producing an AP site by cutting the glycosidic linkage. But this is an alternative mechanism, right? So here we use an enzyme, MGMT, and that enzyme simply undoes by clipping off the alkylating group. That's it. Unfortunately, these enzymes, they work in a stoichiometric fashion. That means, unlike other enzymes, which can go and get recycled at the end of the reaction, they're back to normal. MGMT is one of those enzymes that binds the chemistry permanently. Therefore, the enzyme can get used up. So unless there's a replacement enzyme being produced by those cells, the system will become saturated. And that's the problem. If you have too much of this alkylating agent present, then the cell becomes saturated and the damage cannot be fully restored to its normal wild type condition. And that has been tested by exposing mice to different amounts of very dangerous alkylating agents, right? So one of those is a methyl nitrosourea. There you go. It's got the so in the middle. This is an alkylating agent, as I said, and in mice that are normal, you can see after a certain number of days, when this chemical is introduced into those mice, they then do develop cancers. But mice that have an extra copy of this gene, they tend to be able to resist, at least at that dose, the harmful effects of MNU. So maybe that's a solution to developing cancer, that we will need to increase the expression of certain types of proteins, enzymes, right? So think about that as a possible therapy for the future. The last panel here, panel D, simply says that lipid molecules, yes, fats, lipid molecules can also become highly reactive. And this is particularly true in inflamed regions of the body. So if you have chronic inflammation, such as with smoking or some type of autoimmune disease, then you're going to be very prone to developing this abnormal lipid molecule. And that then can attack the bases, forming DNA adducts, okay? So naturally, we have some systems in the body that are able to use enzymes that are able to use their activity to restore the chemistry back to normal. So you can see this part of the molecule is removed, restoring the base back to where it should be. So you'll see the examples given on these pages are all talking about various mechanisms that you should be familiar with. If you go back to the slideshow, we put these in a chronological, well, not chronological, but we put them in an order together, so that makes sense. It's not chronological. So here we have a mechanism, a DNA repair mechanism called BER, base excision repair. And what they're doing is they're showing us how a base that's chemically altered using the mechanisms that we just mentioned in the previous couple of figures, how they can then be corrected. So here the base is cleaved, just the base, and then the backbone is excised, right? So it's not possible to replace the base without putting the nucleotide back. So one way is to just replace the nucleotide. So that's called the short patch repair. An alternative mechanism that exists, because there's multiple repair mechanisms running around, would be to do a long patch repair, where the DNA is cut a significant distance apart, and a patch of backbone is removed, single-stranded DNA is removed, and then repaired in its entirety. This is called flap endonuclease. You would have come across FLAP in your genetics course. Okay, so let's highlight that as something important. So there's FLAP, endonuclease, right? An alternative mechanism that looks for alternative types of damage is nucleotide excision repair, NER. And again, NER occurs in two varieties, NER-A and NER-B, and they do talk about that in the actual copy of this chapter. So here we have an adduct, something added to the DNA, some kind of distortion in the DNA backbone, and that is then detected. And now we're going to cleave a large segment of DNA, about 30 to 100 bases, right? And then that is then filled in by the repair polymerases using regular DNA replication methodology, okay? So two mechanisms. One deals with one type of damage, the other one deals with an alternative type of damage. The book then goes on to talk about bypass synthesis. So bypass synthesis is an alternative fails safety mechanism. So when none of the other systems are able to correct the damage, then bypass synthesis comes along and says, okay, we have to get over this damage. So I'll take a guess across the lesions of DNA. At least we can get the replication fork going again. So that's just taking a chance. Sometimes you guess right, sometimes you guess wrong, right? And that is error-prone repair. So that comes under the title of error-prone repair. And we have special bypass polymerases which are manufactured, and they're the ones that don't have a proofreading function, but they can polymerize and they can see the damage in front of them on the template, and they can somehow hop over that damage and get through it at least. And you can see some of the consequences that there are alternatives because of the guessing of the system. Sometimes it produces the right outcome, and sometimes it produces a more mutant outcome. But it's better than stalling the DNA replication machine, at which the cell will die regardless. All right. So exposure, remember, we're still looking at exogenous cases in most of these, although endogenous cases can also be treated by the repair pathways. So here we have a classic example of leathery skin of a patient that has a xeroderma pigmentosa. Xeroderma pigmentosum, right? And these people have a defect in the NER repair pathway. And you can see they're more susceptible to DNA damage. The same is true of people that have red hair. That red hair is correlated with less production of melanin, and therefore there's less protection from ultraviolet radiation. Okay. So let's continue at the bottom with section 9, which is now inherited defects in nucleotide excision repair, or base excision repair, or mismatch repair. So these are the three major mechanisms, NER, BER, and MER, all right? And you can see there's a correlation, direct correlation between the percentage accumulation of damage and the age of the patient. So for most of us that don't have this genetic alteration, we follow the green line. But for patients that have XP, they follow the red line. So these are pretty devastating mutations to possess. So how do we discover the number of genes involved in each repair process, in each repair mechanism? We did classical genetics in those days. We looked at two patients. We took their cells, we merged them together, or we used mice models, and we were able to use complementation testing, complementation analysis. Simply put, if mouse 1 has a defect in one gene, and mouse 2 has a defect somewhere along the same gene, and then you make a hybrid cell, as you can see here, because the defect is in the same gene, now you have two defects in the same gene, it's not going to produce the enzyme. Therefore, the mouse is still susceptible. In the second example on this side, if you have a mouse with a defect in this enzyme, and you have another mouse with a defect in that enzyme, and you make a hybrid cell, guess what? Because each cell is able to produce the second enzyme normally, for instance, if this mouse has a defect in enzyme A, but enzyme B is okay, their pathway is still broken because it's a pathway. And then over here is the opposite, right? They're normal for enzyme A, but they have a defect in enzyme B. So that pathway is jammed too at that point. You put them together, guess what? The two normal copies, A and B, are able to complete a full pathway. Therefore, the mouse becomes resistant to the assault. So because the two mice have genes, two separate genes that are controlling these two enzymes, then when they're put together, they complement each other. One makes up for the loss of the enzyme in the other, in both cases, right? Therefore, you restore a full biochemical pathway. So that's what complementation testing does. It's a genetic way of discovering if the mutations are in the same gene or they're in different genes of a pathway. So by doing this type of analysis, we have a better understanding of the number of genes that are involved in these various repair mechanisms. So if you read this, you can see down here, we have 7 or 8 genes that are associated with XP repair, right? And you can see other examples further down. So complementation testing was useful in determining how complex each repair pathway was. What we'll do now is just look through this table, and then we'll stop and have another break. So table 12.1 simply looks at inherited cancers, right? So you'll get used to this word because they're going to use it a lot in this textbook. Familial cancers simply means at least one of the genes that you require for something is being passed through from your parents in a mutated form already, okay? And you can see many different syndromes associated with cancer have their genes identified, and the actual biochemical mechanism responsible has been elucidated. So this is our progress towards understanding cancer and then figuring out what and where things go wrong. So thank you for now, and we'll start on the next figure in the next video. Thank you. Bye.